Hello everyone and welcome back to Physiology. In this video, we're describing the cellular events that lead to contraction and relaxation of smooth muscles. And once again, we'll compare what happens in the smooth muscle cell to what we already know happens in the skeletal muscle cell. One of the differences that we will highlight here is that smooth muscles can actually contract in response to different types of signals. It can be electrical, like an action potential. It can be chemical, such as a signal molecule, or it can be mechanical, such as stretching. So here is our working map of how smooth muscle contraction occurs. We have 6 steps here. It starts with a stimulus, which then creates a calcium signal. This calcium signal is going to phosphorylate myosin light chains, and that will directly lead to myosin ATPase activation. Once the myosin ATPase is activated, actin and myosin can interact into the cross-bridge cycle, and that leads to myosin pulling on actin, which we know is called the sliding filament theory. Now, there's different colors here to highlight that anything in yellow here is specific to smooth muscle. So these are events that are different than what we described in skeletal muscle. In red here are characteristics that smooth muscle cells share with skeletal muscles. So in the next few slides, we will talk about steps 2 to 4. And we'll answer the questions, how do the calcium signal lead to phosphorylation of the myosin light chain, and how does that lead to the activation of the myosin ATPase? So I first want to bring your attention to the cartoon on the right here. So what we're looking at is the extracellular fluid here, and then the intracellular fluid or the sarcoplasm of the smooth muscle here. So the intracellular cascade that leads to the contraction of smooth muscle starts with calcium. So where does this calcium comes from? The calcium that starts this process actually comes from 2 different locations in smooth muscle. The first one is from the extracellular fluid. So you see here that calcium from the extracellular fluid can come inside the cell through channels when they're open. There's also some calcium that comes from the sarcoplasmic reticulum. And we've seen this when we talked about skeletal muscle. So in smooth muscle, calcium actually comes from 2 different places. Most of it comes from the ECF or the extracellular fluid, and some comes from the sarcoplasmic reticulum. And this is what I tried to highlight here. The 3 pluses here just refers to the fact that the majority of the calcium comes from the ECF, and there's just a bit of calcium that actually comes from the sarcoplasmic reticulum. If you think back of our skeletal muscle knowledge, all of the calcium in skeletal muscle comes from the sarcoplasmic reticulum. So this is a bit of a difference. So when calcium moves, both from the extracellular fluid and the sarcoplasmic reticulum into the sarcoplasm, the concentration of calcium inside the sarcoplasm increases. And when that happens, calcium binds to a molecule that we call calmodulin, or CAM. And we see the CAM right here. So when calcium is low in the cell, calmodulin is by itself. When calcium concentration increases, then we have this complex of calmodulin that's bound to calcium, which we have right here. So now this complex is important because it takes an enzyme that we call the myosin light chain kinase, or MLCK, from an inactive form to an active form. So here we have the inactive myosin light chain kinase, or MLCK. When this enzyme encounters the complex of calcium and calmodulin, it becomes active, which is what is shown here by this arrow. We turn an inactive MLCK into an active MLCK. Now let's go back to the name of this enzyme. It is a kinase. Remember that all kinases in our body do one thing and one thing only, is to phosphorylate a protein, or to add a phosphate group to a protein. So this kinase is no different. It will phosphorylate something. What will it phosphorylate? It will phosphorylate the light chains of the myosin. And that's what we see right here. So you have the myosin, and you recognize it's the heads of the myosin. When MLCK becomes active, it adds phosphate groups that are highlighted here in green onto the light chains of the myosin. And that's what will activate the myosin heads and therefore stimulate the cross-bridging. So this last part here is a cross-bridge cycle that we're already familiar with. It's when the head of the myosins interact with actin. So I know this was a lot of information. There's a few slides coming up that will give us a little bit more details about what we've talked about here. And then we'll come back to this slide for a review and a summary. First of all, let's unpack or review what myosin ATPase is. So this is a slide that we've already seen that is an overview of the cross-bridge cycling. What's especially relevant in this case is how the myosin head binds ATP. So if you remember, the myosin head has a binding sites for nucleotides, so either ATP or ADP. And we know that the binding of ATP is required for the head to dissociate from the actin. Now the step right after that is that there's the myosin ATPase, so there's an enzyme inside the myosin head that is able to hydrolyze ATP or break down ATP. So that's when the myosin ATPase takes ATP and breaks it down into ADP and phosphate. And that provides the energy to reposition the head for the power stroke. So we see here the head of the myosin has changed its angle. So it's no different in smooth muscle. The take-home message here is that the myosin ATPase activity dictates how quickly the cycle can happen. So it determines the cross-bridge cycling rate. So if we think back about things that happen in a smooth muscle, when myosin ATPase activities increase, this cycle can go on quite quickly. When myosin ATPase activity decreases, then this cycle either slows down or stops, and that decreases the force within the muscle cells. The second piece of information that we need to fully understand smooth muscle contraction is what are myosin light chains. So what we've said so far is that calcium binds to calmodulin. And this complex activates the myosin light chain kinase. And that this myosin light chain kinase phosphorylates the myosin light chains. But we haven't really talked about where are myosin light chains and what they do. So hopefully this cartoon can help us answer this question. So what we're looking at here is a myosin molecule. You can recognize the head here with its two binding sites. So binding sites for ATP and a binding sites for actin. You can also recognize the neck or the hinge region of the myosin and then its long tails. But what I really want to bring your attention to is these two molecules that are associated with myosin. These are the light chains. So there's actually two light chains. One is called the essential light chain, which is shown here in yellow, and the second one is the regulatory light chain, which is shown here in red. And that's where the myosin light chain kinase phosphorylates or puts the phosphate group. So as you can see in the bottom, when the light chain has no phosphate group, the myosin ATPase activity in the head is slow and muscle tension is also low. Now, when the myosin light chain kinase adds a phosphate group to those light chains, that increases the activity of the myosin ATPase, and that also increases muscle tension. All right, so let's bring this back together and review all of this information. In smooth muscle, the cascade that leads to the cross-bridging always starts with calcium. Calcium comes primarily from the outside of the cell, or the extracellular fluid, and a little bit from the sarcoplasmic reticulum. When we have lots of calcium into the sarcoplasm, it will bind to the calmodulin molecule. And this calcium-calmodulin complex turn an inactive myosin light chain kinase into an active myosin light chain kinase. A kinase always adds phosphate group, so it will phosphorylate the light chains that are around the neck of the myosin molecule. And this will lead to an active activation of the myosin ATPase and therefore stimulate the cross-bridging and increase muscle tension. So hopefully now we have a better idea of how this happens. And I really do encourage you guys to try to reproduce this cartoon in your own way, in your own words, so that you can put all of these steps in order in your head. The next question is, how does smooth muscle tension decreases? In other words, what needs to happen in order for smooth muscle to relax? There's two main things that must happen. The first one is basically a reversal of everything we just talked about. And it starts by reducing the amount of calcium inside the cell. To do that, calcium must be actively transported out of the sarcoplasm, right? Actively, because it will be transported against its concentration gradient. So calcium will be pumped back into the sarcoplasmic reticulum via calcium ATPase. And then calcium will also be pumped back into the extracellular fluid via a very similar calcium ATPase. And there's also a second molecule, an antiport, that will transport calcium out of the cell in exchange for one molecule of sodium inside the cell. But the take-home message here is that there are a number of transporters that will move calcium from the sarcoplasm back into the outside of the cell or back into the sarcoplasmic reticulum. And this means that the concentration of calcium inside the cell decreases, and that will reverse all of the actions we just talked about. When calcium concentration in the cell is low, calcium unbinds from calmodulin, so calmodulin is on its own again. That will inactivate the myosin light chain kinase, therefore decrease myosin light chain phosphorylation, and the end result of this is that it will decrease the myosin ATPase activity and decrease muscle tension. So the first way that tension decreases in smooth muscle is by reducing the amount of calcium inside the cell. The second way, and they actually work together, is via an enzyme that we call the myosin light chain phosphatase. So we're now very familiar with the myosin light chain kinase, the K for kinase, that adds a phosphate onto a protein. Now we have the myosin light chain phosphatase. And all of the phosphatases in our body have one function, is that they take away phosphate groups. So as the name says, this myosin light chain phosphatase, or MLCP, actively takes away the phosphatase on the myosin light chain. And that leads to the dephosphorylation of the myosin light chain. And that's why we have this arrow going the other way. We take the myosin light chains that had a phosphate group, and now they no longer have a phosphate group. And that also contributes to decreasing myosin ATPase activity and decreasing muscle tension. So these two mechanisms will work together and take place at the same time in order to coordinate smooth muscle relaxation. So the last few slides have talked about our steps 2, 3, and 4, but we haven't yet talked about number 1 yet, which is what are the stimuli that lead to smooth muscle contraction. So there's two main types of stimuli, electrical and chemical. So the question we're answering here is which stimuli initiate this calcium signaling? We know that calcium must get into the cell, but we don't know what's the initial stimulus that will open those channels to let calcium in. A very important characteristic of smooth muscle here is that they will respond to diverse stimuli. And that really depends on the type of receptors that they express. If they have voltage-gated receptors, they will respond to electrical stimuli. If they have ligand-gated receptors, they will respond to chemical signals. We will also see in the next few slides that smooth muscle contraction is a graded process that is proportional to the amount of calcium in the cell. So in other words, the greater the calcium in the cell, the greater the tension is created. So as I hinted in the previous slide, calcium enters smooth muscle via different types of membrane channels. Some of them are voltage-gated, some of them are ligand-gated, and some of them are stretch activated. So if we go back to the cartoon that we've talked about here, we're talking about this channel. So that's the channel on the plasma membrane of the smooth muscle. This type of channel changes depending on the type of smooth muscle cell that we're looking at. So if this channel is a voltage-gated calcium channel, it will open in response to depolarization. But this channel can also be ligand-gated. And in this case, that channel will only open when the appropriate ligand or the appropriate chemical signal binds to the channel. And finally, there are some channels that we call stretch activated. And those channels open when pressure or force distort the cell membrane. Now I have a question for you. Let's say that this channel here is a ligand-gated calcium channel. So there's a chemical signal that binds to this channel and that allows calcium to move through this channel and enter the cell. What happens to the cell membrane potential when calcium comes in? Remember that calcium is a cation or a positive ion. So when calcium ions enter the cell, it actually depolarizes the cell. That leads to cell depolarization. And when the cell becomes depolarized, it can activate voltage-gated calcium channels. So that's why I have here a note that calcium entry from either number 2, ligand-gated channels, or number 3, stretch-activated channels, can activate your number 1, your voltage-gated calcium channels. So this is just one example to show you that a cell can express different types of receptors. It could have, for instance, ligand-gated and voltage-gated channels, and that these channels work together in order to move calcium inside the cell. The second question we have to answer is, how do we get the calcium from inside the sarcoplasmic reticulum to the cytoplasm? In other words, how does this channel open? So once again, this channel can actually be different types of channel. The first one we will describe is one we've already talked about. Let's say this is our ryanodin receptors, or RYR. The ryanodin receptors actually has a calcium binding site. So when calcium comes in, I'm going to get some calcium here in green. So when calcium comes in from the extracellular fluid, some of this calcium can bind to the ryanodin receptor. And when calcium binds to the ryanodin receptor, it opens and therefore allows more calcium, this time calcium from the sarcoplasmic reticulum, to enter the sarcoplasm. So that's what we called calcium-induced calcium release. If you break down this term, it literally tells you that the calcium released from the sarcoplasmic reticulum is due to a calcium signal. So once again, that happens when some calcium enters from the extracellular fluid, binds to the ryanodin receptor, opens the channel, and allows calcium to exit from the sarcoplasmic reticulum. So that's the first way that we can get the calcium from the sarcoplasmic reticulum. The second way is called IP3 mediated calcium release. So as the name says, now the signal molecule that will open this channel is not calcium, but is IP3. So here, this channel would be an IP3 channel, and it would work relatively similar, where when IP3 is present in the cell, it can bind to the channel that allows channel to open and calcium to exit the sarcoplasmic reticulum. Now the question is, do we remember how IP3 actually gets into the cell? Here's a cartoon that we've already seen when we describe cell communication. So the phospholipase C pathway, or PLC pathway, is the main pathway that produces IP3, which is a second messenger. So as a reminder, it happens when the G protein receptor is activated and then activates the phospholipase C. Phospholipase C then produces two different second messengers, diacylglycerol and inositol triphosphate, or IP3. In this case, we're interested in the IP3. So the IP3 binds to the channel on the sarcoplasmic reticulum and allows calcium to get out. And that's what's written right here in the previous slide. Your G protein coupled receptor is activated. That activates phospholipase C and produces a second messenger, IP3. IP3 binds to these IP3 receptors on the sarcoplasmic reticulum, and the channels will open and calcium will move outside of the sarcoplasmic reticulum. So here are the two ways that we can get calcium mobilization from the sarcoplasmic reticulum. This channel in the sarcoplasmic reticulum either opens in response to a calcium signal or in response to an IP3 signal. To illustrate even more that smooth muscle can respond to very, very different signals, let's look at this graph here. So this cartoon shows tension in red versus membrane potential in blue. So if we first focus on the top panels here, you can see in blue here that this cell is firing action potentials. And at the same time as the cell's firing action potential, we also see the tension in that same cell increasing. And we see this in red here. So this is not different. This is something that we're comfortable with, where action potentials lead to an increase in tension. So we see this here. We also see this in the right panel. Action potentials lead to an increase in tension. Action potentials also lead to another increase in tension. In this right panel, we can also see another characteristic that we're familiar with. The higher the frequency of action potential, see here the frequency of action potential is greater than here, it leads to greater tension. So high action potential frequency, F here for frequency, leads to higher muscle tension. And you can see here that these action potentials have different names. So on the left-hand side, we call them pacemaker potentials. On the right panel, we call them slow waves. And we'll see what they are in the next few slides. But before we go there, I also want to bring your attention to the bottom of the slide here. In this left panel here, we see that the membrane potential changes a little bit, but never fires action potential. This is what we would call subthreshold depolarization. So the cell is depolarized a little bit, but it hasn't reached threshold, therefore there's no action potential. In smooth muscle, it doesn't matter. You can see that the tension of muscle that we see in red here mirrors the depolarization and the hyperpolarization, even though it doesn't reach threshold. So when the cell hyperpolarizes, the tension decreases. Conversely, when the cell depolarizes, the tension increases too. So this is to show you that smooth muscle contraction can occur even when the depolarization do not reach threshold. Let's take this a step further. Here is a cell where the membrane potential is stable. There's no depolarization, there's no hyperpolarization. Yet, you can still see that the tension can change. It can increase here, it decreases here. Why is that? In this case, it's because the cell is responding to a chemical signal. So chemical signal X here is applied, and that leads to depolarization. And when chemical signal Y is applied, it leads to hyperpolarization. So I feel this is a really good cartoon that shows us how varied the signals can be in smooth muscle. From action potential to subthreshold depolarization and even stable membrane potential can all lead to change in tension in smooth muscle cells. Now let's go back to our pacemaker and our slow waves. So a lot of smooth muscle cells have spontaneous depolarization. Slow waves and pacemaker potentials are two of those types of spontaneous depolarization. So let's start with slow waves here. What we see here is on the y-axis is the membrane potential, on the x-axis it's time. So we're looking at depolarization of a cell here. You can see that it's cells that have slow waves. The membrane potential goes up and down and up and this is spontaneous, meaning that those cells do not require an external stimulus for the membrane potential to change in that way. Now, what's unique to slow waves is that when an additional stimulus occurs, the slow wave will actually reach threshold and then fire action potentials, which you can see here. So in this graph, we're looking at 3 different slow waves. The first 2 do not reach threshold and do not fire action potential. The last one, because of additional stimulus, does reach threshold and fire action potentials. So in this case, only the third slow wave, only the one that fire action potential, will actually lead to contraction. Let's compare this with pacemaker potentials. So cells that have pacemaker potential also depolarize spontaneously without the need for additional stimulus. But in the case of pacemaker potential, their characteristics is that they always reach threshold. So depolarization reaches threshold, action potential. Next depolarization, action potential. Next depolarization, action potential, and so on and so forth. And every time that this action potential occurs, there's a little bit more calcium that comes into the cell, and the concentration of calcium inside the sarcoplasm increases. And that leads to smooth muscle contraction. And finally, as we said, ligands or chemical signals can increase or decrease smooth muscle tension without any change in membrane potential. Here's a few examples of ligands that will either contract or relax your smooth muscle cell. Epinephrine and norepinephrine in some tissues lead to contraction. Epinephrine and norepinephrine in other tissues lead to relaxation of smooth muscle. Smooth muscles can also respond to acetylcholine, nitric oxide, or histamine. And these are only a few examples of chemical ligands and neurotransmitters or paracrine signals that can contract and relax smooth muscle. Now I have a question for you. We see here that the same ligands, let's take epinephrine for instance, have different actions on different tissue. So epinephrine on your blood vessel will contract the smooth muscle there. Epinephrine on your bladder will relax the smooth muscles here. How can this happen? So this should be a review of our concepts about cellular communication. It happens because the tissues express different receptors. So the blood vessels would have different receptors than the bladder, for instance. So when epinephrine binds to the receptors in blood vessels, it contracts the smooth muscle. When epinephrine binds to a different receptor on the bladder, it leads to the relaxation of smooth muscle. Here are a few summary slides to make sure that we picked up on all the important take-home messages from the smooth muscles. The first table here will help us compare smooth muscle to skeletal muscle. So do smooth muscles have sarcomeres? No, we already know this. Calcium comes from the extracellular fluid mostly, and a little bit from the sarcoplasmic reticulum. Which protein does calcium bind to? In skeletal muscle, it's troponin. In smooth muscle, it's calmodulin. Can contraction start with an electrical signal? Absolutely. Can contraction start with a chemical signal? Yes, as well. That's different than in skeletal muscle. Our twitch length is slow. Your smooth muscles do not have T-tubules. And contraction can start without an action potential. And finally, the speed of the myosin ATPase is slower than in skeletal muscle. Our last summary slide here, guys. So in smooth muscle, actin is attached to what structure? Do you remember? They are called the dense bodies. Dense bodies. Sorry, I couldn't couldn't actually write on the line there. But actin attaches to the dense bodies and they surround myosin to ensure that contractions occur no matter the shape of the cell. Similar to skeletal muscle, cross-bridging leads to the sliding filament model and to the contraction of the smooth muscle. Calcium comes from the extracellular fluid and the sarcoplasmic reticulum, binds to calmodulin, which activates the myosin light chain kinase. Okay, that was a new molecule for smooth muscle physiology. Smooth muscle relaxation required the active transport of calcium outside the cell in or back to the sarcoplasmic reticulum, and the activation of our myosin light chain phosphatase, right? That enzyme that takes phosphate away. Going down to here, smooth muscle can be single unit or multi-unit. And the single unit smooth muscle have gap junctions. And that's a key characteristic that allow those ions to move rapidly from one sarcoplasm to the other. Smooth muscle can contract due to action potentials. They can also contract in response to subthreshold depolarization. They can contract in response to chemical signals, and they can contract in response to stretch. And that's because cells can express a variety of calcium channels. Calcium can enter the sarcoplasm from the extracellular fluid through ligand-gated, stretch receptors, or voltage-gated. And finally, calcium efflux from the sarcoplasmic reticulum is either due to calcium binding to the ryanodin receptor or our second messenger IP3 binding to its specific IP3 receptors. That's it for smooth muscle physiology, guys. As always, if you have any questions, don't hesitate to drop by our office hours or to send us an email.